Finally today, last week, Nebraska Research Assistant Professor Katja Koehler-Cole discussed her research with cover crops in corn. This week, she's back to discuss a similar project, but this time in soybeans.
We've been growing cover crops in these plots since 2014, and one of the things we're interested in is how cover crops influence the abundance and diversity of soil microbes. If you can look down here, this is a plot that is a control plot, so we have not grown a cover crop in here. The top layer of the soil is pretty dry. We've had a very dry August. The top layer of the soil is also where most of our soil microbes live. They're probably in the top 3 to 4 inches is the majority of the soil microbial life on Earth. So it does make sense to give them, if we can, the best possible growing conditions, which would be moderate temperatures and food and shelter, of course. And cover crops are one reason that we can do that in our crop fields. This was a cereal rye cover crop that was terminated in early May of this year, and then soybean were planted right away. It was basically a green planting. So you can still see a lot of the cover crop stems. It's noticeable that almost all of the soil is covered. That made a big difference, especially at the beginning of the season when the soybean are still small. So that residue really helps prevent erosion. It keeps the soil cooler and more moist. So cover crops are especially useful if we're growing them during a time when there's no other crop in the soil. So in our corn-soybean rotation, that would be after the harvest of, for example, corn until the planting of the next corn or soybean crop. And winter cover crops such as cereal rye or hairy vetch are both pretty useful. When they're in the ground, they provide food to microbes. Microbes' preferred food source are the simple sugars and amino acids that are leaking from plant roots. If we have a living plant in the ground, we're providing both food and shelter for soil microbial communities. We're using a test called phospholipid fatty acid, or short Plfa analysis. We're having it analyzed at a local lab. For this test, we're just taking the upper 4 inches of soil, and this is really difficult right now because the soil is very hard. I'm putting this in a Ziploc bag. The soil has to be either taken to the laboratory right away, or if you can't do that, it's best to freeze it. This Plfa test gives us some idea about the different microbial groups. They are the ones that break down simple organic materials such as fresh leaves. If soybean leaves fall down, they can easily degrade that, or if we have a fresh young cover crop, they are degrading things like that. Fungi are another group that we are interested in and that we are hoping to increase with a cover crop. Saprophytic fungi are the ones that are the decomposers. And another important group of fungi are arbuscular mycorrhizal fungi, or AMF fungi for short. So they help plants access nutrients in the soil, such as phosphorus or even water, that the plant roots themselves can't get to. And they can colonize, for example, a cereal rye cover crop. And then when a corn crop is planted, they can go from the cereal rye and colonize the corn crop. So really, with a cover crop, we're kind of providing sort of a bridge or an alternative host to the AMF fungi. What all fungi do is they're hyphae, they're very thin, thread-like structures. They reach out in the soil, they are the ones that, forage for nutrients, and they excrete a glue-like substance called glomalin, which helps bind soil particles together and form soil aggregates. In our research in these plots here, we've been growing cover crops continuously for 4 to 5 years now. The soil is very dry. Again, we had a very dry August, but we have found in our research that we have seen improvements in soil aggregation. Cover crops can help our microbial communities, which in turn is very important for our soil structure.